Hello, everybody. Hello, hello, hello. Can see lots of people coming into the webinar room. It's great to have you here today. If you can do me a big favor and let us know if everything looks good, if everything sounds good, if everything is working well for you. If you can go to the chat and just pop in your name and where you are listening to us from, that would be really, really good, so we can see that everything's working. We can also see that the chat function is working, because this is where we want to get all of your questions around today's webinar. So just go ahead, tell us who you are, where you're calling from, so we can get a good picture of who's listening to us today. Just looking over the chat there. We've got Stockholm, fantastic. York, fantastic. So just keep those coming in so we can check. Also, of course, let us know if there are any technical problems that we can fix as we go along. But to give you a little bit of an introduction to who we are and why we're doing this, this presentation, this webinar today, my name is Ben Liverton, talking to you from Ebba Biotech, and we're a Swedish company started in 2015. We focus a lot on and we care a lot about interdisciplinary collaborations, and I think if you've seen any of our previous webinars, you'll see what we mean by interdisciplinary. We cover lots and lots of different topics. Our products are Optotracers, and these are fluorescent tracer molecules for labeling macromolecular components, and that is exactly what we're going to hear about today. So today, it is my great pleasure to introduce to you Professor Ulrica Edlund, and she is a professor in Polymer Technology at KTH, as well as being the Vice Director of the Research Consortium AIMS, which is the Centre for the Advancement of Integrated Medical and Engineering Sciences. She has a passion for finding sustainable solutions to maximise the potential of natural resources, and she focuses on developing sustainable methods to bring forward new green materials from renewable resources such as films, coatings, hydrogels from non-cellulosic fractions of wood processing, as well as lots and lots of other cool things. And we're going to hear about some of her research today. Everybody, it is my great pleasure to now hand over to Professor Ulrica Edlund.
Thank you so much, Ben, and thank you everyone who's tuned in to listen to a story. It's a great pleasure to share some of the work that we're doing on applying the concept of optotracing to understand and sort of evaluate how we are dealing with biomass and how we can make the most of nature's biomacromolecules, both in terms of recovering them from nature and also converting them into functional and advanced materials. So I work at Fiber and Polymer Technology at the KTH Royal Institute of Technology in Stockholm, Sweden. And I'm a proud member of AIMS, the center that Ben just mentioned, affiliated also with Karolinska Institute. And this is where this work is located, in the borderline between KI and KTH, sort of connecting competences that makes us grow further beyond what we could do with our own expertises. We grow together. So my work is focusing on how to utilize biomacromolecules, polysaccharides in particular, but also other molecules in nature as resources for building materials. So we're looking into what molecules that nature already provided, the context or the tissue, the complex construction of these tissues where the molecules are located, trying to understand how we can possibly fractionate and recover these molecules, preferably by recovering them without destroying or detrimentally altering the structure of the others, so that we get a whole range of different molecules. So putting up these biorefinery process is a big part. Then, of course, looking into what different fractions we get out, what structures are hidden in the biomass, how are they connected to each other, can they be separated, should they be separated, how do they work, how can we modify them, how can we impart advanced functions that are not already there, how we can combine them into materials with different property profiles. How can we use those materials? What could they possibly be good for? Can we replace what is today made with fossil-based materials to increase sustainability in the material usage practice in society? How will we then make a specific product, geometry, shape, function from that material? We also care, obviously, about the post-consumer perspective. When I'm done using my product, it's not a waste. It's a bunch of raw materials for the next round in a circular bioeconomy. So we also have to look at the chemistry post-use. How can we make use of that? How can we disintegrate that? That's another type of biorefining, to valorate the molecules that we have in waste and make them a feedstock for the next generation of materials. So we work in the entire value chain, and there are many different aspects, of course, that come into play here. But essential, wherever you are in this loop, is to understand what you have. So characterization, that's the essential part. And this is where Optotracing turned out to be the most valuable tool that we have to understand our structures and know where they are and where they're going. We make a lot of different materials from polysaccharides. I mean, some argue that there is an advancement in the fossil-based materials, that it can do many new things. But we claim that many or even most of these things can also be made with bio-based polymers, that you can find the same type of function and that you can modify the function so that you get whatever property profile that you're looking for. So this is just a little menu of the different materials that we have made from polysaccharides extracted from various biomasses. But it is also challenging, of course, to work with biomasses. It's a quite complex structure, many different types of structures depending on which particular plant you're looking at. So to start such a value chain, you sort of have to know, at least have a rough idea of what your biomass is containing and how these different molecules fit together. And from there, make a qualified guess on how could I potentially recover the molecules and separate them, liberate them from the matrix. So we have to try different process parameters and settings to try to fractionate our molecules. Of course, we have to compare the outcome from different types of processes to see which one was more feasible, which one did actually give us a whole feedstock of different molecules without destroying the others. How can I make the most of my biomass so that I don't discard half when looking for a target molecule? So analysis will be core here. And this is where we run into a lot of challenges, because the methods that we have at hand are typically destructive and also not conclusive in actually telling us what structures we have. Most of these analysis methods, they rely on us first deconstructing the native polymers and low molecular weight stuff that we have, and then telling us what that deconstructed material consists of. And that gives us many clues, but it will not tell us exactly how these building blocks or the deconstructed fractions were fit together in the native structure. So we just have to guess. So what I'm showing you here is a very common way of analyzing your polysaccharide fractions, ion chromatography, where you first break down your polysaccharides into the monomeric sugars, then you analyze which sugars you have, and then you try to backtrack where could those molecules possibly have been linked together in the native biomass. It's all about guessing, of course. And from all the information we get, we can now go back and iterate the loop, trying to do better in terms of fractionation, looking at the new type of fractions, what did we get out now, and improve the process until we have something that is feasible and gives us a very useful and functional set of molecules that we can then use as feedstock. So a better menu of analysis methods would really ease things for us. And this is where we look with some envy, I have to admit, at those who work with proteins, because what they have are staining techniques that selectively can stain and show where different proteins are located in tissues. There are antibodies that fits the different proteins. So if you just know which to target, you can probably find an antibody kit. You can even buy a lot of kit assays, where then you can stain and verify that you have a certain composition. Also, this can be non-destructive. This is the type of analysis technique that we would like for our polysaccharides, but we do not. What we have are structural analysis methods like FTIR that for sure will tell you that you probably have a polysaccharide and a little bit about how it's functionalized, but that is poor in discriminating one fairly similar polysaccharide from the other. Trying to discriminate, for instance, amylose from cellulose would be very difficult. Our standard tool would be ion chromatography, as I just mentioned, in which you first deconstruct the polymer down to the monomeric sugar state, analyze which sugars you have, and you get sort of a receipt on the building blocks that you have. And then you have to guess, how were they linked together before I destroy them? And of course, if I'm now working with a sample containing both starch and cellulose, both being homopolymers of glucose, well, then I will end up with the result as you see here. Okay, a lot of glucose. And I have no clue in whether these came from starch or cellulose or both. And if so, in what share I have cellulose to amylose. So we're sort of limited in the current analysis techniques. NMR can get you quite far, but also it can be non-conclusive. So this is what we have. This is what we want. And this is where we're stuck. Microscopy can tell us a lot about the morphology. We can zoom in and get really detailed structure information about the matrix morphology and topography, but it does not tell you where different molecules are located. The staining techniques, if we borrow those from protein science, are definitely not going to help us at all. They're not selective, they do not bind, and it leaves you with a very fuzzy image of something that you cannot really understand. This is where we are. This is what we want. And what we have found in our work is that optotracing can do this for us. So optotracing, if you're not familiar with the concept, is about using molecules like these, optotracers, which are basically oligomers of thiophenes, making a conjugated structure. Can be maybe 5, 6, or 7 units linked together. And of course, you can play around with the functionality. You can add heteroatoms like sulfur. You can have different side groups and so on. What they have in common is that they have a really interesting optical behavior. They can dynamically change their composition and thereby change the optical properties. So they can be sort of excited with light within the visible spectrum. And that means that we can see them. They will emit fluorescence, so we can detect that. But we also see that they emit at different wavelengths depending on their morphology, or rather their structure. So if they're free, they're quite loose and they can move around, and you get one type of optical response. But if they are to bind to, say, a planar surface, they then will adapt their conformation and line up and interact closely to that surface, meaning that the morphology or the structure of that optotracer has changed. And this change you can actually detect. You can see it as a shift in the maxima of the emission. And you can also see that you typically get a higher intensity of that emission. So we both have a shift, a red shift in the spectrum, and we also have a shift in the intensity of that response. And sometimes you also see spectral sub-patterns, like you can see here, where you have shoulders on each side of the sample. So this is interesting. If we can now make these optotracers interact with the solid surface of a molecule, well, then you can detect when it binds, and that could give you an idea of what it binds to and how much it does. So this has been used quite a lot to trace protein aggregates. And there are many, many publications on how it can be used as such, not in the least for tracing protein aggregates that could be an early sign of Alzheimer's disease. What we sort of hypothesized early on in our work is that probably these Optotracer molecules can also bind to polysaccharides. And if so, can we use them to label polysaccharides and detect them with fluorescent spectra? So we started off with looking at cellulose. If you work with plant biomass and are interested in polysaccharides from nature, well, you're bound to come across cellulose. It's fair to say that it's the most abundant polysaccharide on this planet. So I work with it a lot. So it's interesting to start right there and see if an Optotracer molecule might be able to bind, change conformation, and show that in a changed optical spectrum that we could detect. And yes, it's possible. The microcrystalline cellulose that we had as sort of a model substrate now has very little response in fluorescence. It's not autofluorescent. When it binds to an Optotracer molecule that we tested, we can see a typical redshift, an increase in intensity, and this quite unique to cellulose subspectral pattern with two shoulders around the emission spectra maxima. So that made us really excited. Now, cellulose comes in many different shapes and morphologies. Here are three typical examples of that. From the microscopy picture, we can see the shape, we can see the surface, we can determine the dimensions, but of course we cannot verify that this is indeed cellulose. While treating them or labeling them with our Optotracer, we found that we, in each case, this gets this unique spectral pattern, verifying that it's actually binding to cellulose. So this made us very excited. Can we use now this Optotracer as a tool to detect where we have cellulose? From the spectra, we can detect that we have cellulose, yes. But if you now combine this with a fluorescent microscope, a confocal, well, then you can also see your sample. And you can see it shine wherever the Optotracer binds to the cellulose surface. So now you can really see where you have your molecule in your biomass. This is really interesting to dig down into. And that's what we did next. How can we understand the behavior? How can we use this concept in different ways? First of all, we looked at the resolution. With this microscope, you can also zoom in and you can get quite high resolution, which means that we, by analyzing different pixels here, can also tell us something about the composition at different places of the same particle. What we see here is that we have cellulose in both locations, but that the Optotracer seems to bind differently, whether it's on the middle of the crystal where you have a flat surface to interact with, or towards the end of the crystal where you basically have an edge and the molecule hypothetically then makes a different conformation when it binds. Something else that we learned early on is that lignin seems to quench the signal of this particular Optotracer. We compared pulps from a commercial pulp factory. They make wood pulp for paper and packaging. And of course, in the beginning of such a process, you have quite a lot of lignin retained in the pulp. And then you typically in such a process apply different bleaching processes to remove the lignin and end up with a very high purity cellulose. Now, scanning the different bleaching steps and the pulp that comes out, we can see and we can even quantify how much the Optotracer binds to lignin and quenches out the signal. So now we can no longer see cellulose or barely see cellulose in a sample containing a lot of lignin. Lignin is autofluorescent, so we were expecting a signal. We were not expecting it to quench the signal. But on the other hand, we can now use this as a determination tool to decide how much lignin is there in my biomass sample. So it seems that this can be a tool that gives us a lot of information other than just pinpointing cellulose as such. Now we were excited enough to want to expand the concept. In our early work, we focused on these two molecules. Let's call them prototype molecules. It's a pentamer and it's an hexamer. But we can expand the concept and look to also other Optotracer molecules from the big family. Another thing that we can do is to pimp our microscope, if you wish, to use a microscope with several detectors that can then detect the signal at different wavelength regions in parallel, so that we can get signals from different excitation maxima. And then you can overlay these images and you can get simultaneous information of the contents of different molecules that Optotracer can bind to. So what can we do with this concept, and how do we understand what we see? Well, first of all, we had to ask ourselves the question, how selective is this Optotracer? Yes, there's cellulose, a very regular and linear polysaccharide, no branches. It's beta 1,4 linked, so it's very linear in its conformation. But does it take a full cellulose then to bind? Or would a glucose molecule be enough, or even a dimer or a trimer? To shed light on this question, we probed different chain lengths of oligomers, starting from glucose and going up step by step to larger oligomers. And what we can find here is that you need at least 8 repeating units of glucose before you start to see this unique optical signature of cellulose. So you cannot detect the glucose with this method. You need sort of a segment of a surface for this oligomer to bind to before we see the optical shift here, which is of course very informative for us to know, since we're typically working with complex biomass matrices where you're probably have both oligomeric, monomeric, and polymeric sugar units coexisting in the biomass. So this we learned. We also learned that branching plays a big role here, and that is sort of expected, because if this typical or unique optical signature comes from the Optotracer binding to the surface, well, then it's fair to believe that different branching points and branching groups may interfere with the branching and maybe cause a different binding or a different morphology shift than if you had a flat surface. So to shed light on that, we checked cellulose with different degrees of substitution of a carboxymethyl group. And we see that as we increase the degree of substitution, the more of these side groups we attach, well, the less intensity we have in the signal. So branching plays a big role, and so does the length of the segment that you're probing. Asking ourselves again, how selective is this molecule? Well, cellulose is a homopolymer of glucose, beta 1,4 linked. But if you look to nature, there are many, many polysaccharides that are homopolymers of glucose. Also beta linked, but with branching, is laminarin, where you have a fairly linear backbone and then branches of glucose and glucose polymers sticking out from the channel with 1,6 linkages. We also find a lot of polysaccharides of glucose with the alpha stereoisomer instead, meaning that you have a different stereochemistry in the linkage between one and the other glucose unit. Starch is probably the most well-known example, which is actually a mixture of 2 homopolysaccharides of glucose, alpha 1,4, completely linear amylose, and 1,4 and 1,6 branched amylopectin. There are also many other branched varieties of homopolymers of glucose in nature. I'm showing a few well-known structures here, dextran and glycogen. So question is now, how specific is this Optotracer to cellulose in particular, or can it also bind to other glucans in the same way? Well, let's try. So we started using an excitation wavelength that we know would give the perfect emission from the Optotracer when bound to cellulose. And then we looked at how it would react with all the other types of glucans that it could potentially bind to. And what we find here, quite interestingly, is that the binding is not unique. The Optotracer will for sure interact also with the other polysaccharides, but it will do it a little bit differently, meaning that we will have a little bit of a shift in the excitation maxima or emission maxima, but also to the shape of the molecule. Also, it may be that 500 nanometers is not perfect as the wavelength for the other glucans as compared to cellulose. So that made us realize that if we have now our multi-detector microscope, that we can take in several signals from different spectral wavelengths at the same time, then maybe we could in parallel detect several different polysaccharides. And if we also had the possibility to use different wavelengths for excitation, well, then maybe we can make each molecule behave as its best, depending on which wavelength we used. So let's try that. Let's use a microscope where we can sequentially use different wavelengths for excitation, and then have multiple detectors that can detect the emission spectra at various wavelength ranges. What do we get? Well, what we get is quite amazing. We get a map with different polysaccharides and of course autofluorescent substances in the biomass shining at the same time as you overlay the picture detected from the different detectors. So what we see here is just a slice of an onion. It's non-destructive. We did not tamper. We did not separate the molecules from each other. It's the native tissue with the native location of the molecules. And you can, in high resolution, in detail, see exactly where I have the cellulose, where do I have the lignin, where do I have the chlorophyll. It's quite amazing that it gives us an entirely new way of looking at the biomass beforehand, knowing where my molecules are, which ones they are, and that will give us so much more detailed information, giving us much better opportunities to optimize the following biorefiner procedure to fit the liberation of these different molecules in the best way. We know what we have, and we know how we can separate them from each other. This is another quite exciting picture, I would say. This is a common problem when working with conventional methods like ion chromatography, that if you have several different compounds in your sample that all contain the same sugar, well, then ion chromatography will just show you that you have a lot of that sugar unit. It will not tell you where it came from and how much of that building block came from which different components. With this optotracing molecule, using this multi-detector method, we can now distinguish between the signal coming from starch, amylose and amylopectin, and the signal coming from cellulose, both being homopolysaccharides of glucose, but still with their own unique maxima and shape of the spectrum. So what you see here are starch granules in pockets in a network of cell walls with cellulose. It gives us a whole new idea of how our biomass is constructed and how it looks. So now we are quite excited to use this tool to guide us in every biorefinery process that we are developing. As we go on from biomasses like potato, that is fairly well known. It's known what it consists of and fairly in what ratios these components consist. But what if we go to more unexplored biomasses where there are no hints already or no data already there on what they contain? Then we would need this kind of tool to help us out. And this is a story that I would like to share on how we really built an increased understanding of what we're working with thanks to the Optotracing technology. The interesting thing is, working with plants, you have at least a rough idea if you work with terrestrial plants on what they will contain, because land-based plants are rich in lignin, cellulose, and the family of hemicelluloses. As you go below the surface of water, you find yourself in a whole new kingdom of plants with very different types of molecule compositions. You will find polysaccharides that are nowhere to be found above the surface of the ocean, and you will find also different compositions of these molecules. So working with macroalgae, we came across this abundant type of green algae. It grows natively along the Swedish west coast and east coast in the south. And it's also, as it turns out, quite easy to cultivate, which makes it an interesting biomass to cultivate and sell as a resource for food, for materials, for textiles, to replace fossil-based alternatives. So we joined forces with the marine biologist at Gothenburg University, who does a lot of amazing research on how to cultivate and understand the life and the growth of these algae. And what we and our colleagues at Chalmers University of Technology were interested in is, what can we use this feedstock post-harvest? How can we make use of the molecules and make food, feed, materials, and fine chemicals? So together with Chalmers, we designed different biorefinery processes to make use of the molecules. My interest is primarily then in pigments, in monosaccharides, and in the use of the polysaccharides. We find a really interesting polysaccharide in this algae that is not available on plants on land, Ulva. And this is something that we started to explore. It's quite an interesting thing, not at all like the cellulose and starch structures that I showed you before. It's highly sulfatized to start with. And it contains also different sugar units that is not part of the polysaccharides I showed you before. It also has some really interesting bioactive properties. So it will be very interesting to explore how this material can serve as a material compound. So we designed biorefinery processes. There's just a few examples of what we've been doing to liberate the Ulva polysaccharide, and also how we can make use of the interesting chemistry to build different structures. For instance, we made hydrogels that can swell in water quite a lot, and then do this reversibly, swell, deswell, deswell up on demand, so that it can use it as a sponge or as a very jelly, soft-like, tissue-like compound. So far, we have explored the use of it as an absorbent for heavy metals in water purification. We've also looked into the rheological properties for using this as a gel-like substance. We're also looking into how to use it as a drug delivery matrix for controlled drug delivery. And of course, we're looking into the antibacterial properties of this polymer for use in biomedical applications. So it's a quite exciting polymer, and we're doing a lot of research on it. Aside Ulva, then, we also get an insoluble fraction that looks very much like cellulose. It's fair to believe it could be cellulose because we know also other macroalgae, they are sturdy, they are large, they need a structural component that helps them keep their physical integrity. And cellulose often turns out to be that structural component. So we know from our work with other macroalgae that cellulose is quite abundant. So our first hypothesis, of course, is that what looks like cellulose in the solid residues after Ulvan extraction is indeed cellulose. So we apply our library of different conventional analysis techniques that we have to look into this cellulose. We can defibrillate it quite easily, actually. It does not contain any lignin, down to a nano-sized cellulose material. We find that it has crystallinity pattern, very much like traditional cellulose, although the crystallinity index or the degree of crystallinity is lower than you would find in, for instance, microcrystalline cellulose or non-fibrillated cellulose from wood. Looking into the NMR spectra, we also find that this looks very much like cellulose. Indeed, it looks identical to cellulose references. So is it then cellulose? Let's make the final check according to our traditional method with ionic chromatography, where if we have only cellulose in this fraction, should end up with something 100% glucose. And we did not. In fact, we found that it's free of Ulvan. That is easy to see because Ulvan, as you saw before, contains also other sugar units like hydrochloric acid and rhamnose. We couldn't find any traces of that. So clearly our cellulose is free of Ulvan. However, there's quite a fair share of xylose residues in there. And xylose does not have any place in cellulose. So first, let's see if this is just a contaminant that we can separate easily from cellulose. We tried several different purification protocols, but whatever we did, a fair share of cellulose remained there. So we have to rethink, are we just taking for granted it's cellulose because that's the sort of easiest to believe, that's the low-hanging fruit? Maybe we should rethink it altogether. Maybe this is not cellulose. Maybe it's just anticipated to be. Maybe this is indeed a copolymer of cellulose and glucose appearing like cellulose. How should we tell? Well, we cannot from these results anyway. Let's look to the literature. Did anyone else run into the same problem? As so happens, very few people looked into the structural details of Ulva macroalgae. There are like 4 papers that try to investigate this insoluble residue, and they made basically the same conclusions as we did. There is all the signs of cellulose, but there are also cellulose in there that cannot be explained. One study just hypothesized that this is not cellulose, this is xyloglucan. Another study said that this is cellulose, this is just a contaminant. A third study hypothesized that if this is indeed cellulose and then cellulose is sort of bound to another polysaccharide, then we could selectively degrade the cellulose part by using selective enzymes. And then if we're able to do that, that would be a proof that we indeed have cellulose. So they made a very ambitious and really nice study on that, but the results were somewhat ambiguous. They could degrade, but they could not fully degrade. So it left us with some questions still. The fourth and most elaborate paper, the one that is most elaborate on the use of Ulva and the contaminant, the constituent of Ulva, is this one, where they actually hypothesize this structure of the cell walls of Ulva, where they say, yes, there is cellulose, but we believe that it's strongly linked to a cellulose glucose copolymer, and that's why it's going to be so hard to separate them from each other, because they're so closely linked together. All right, can we, with the Optotracer tool that we just developed, shed light on this question? If there is cellulose, then the Optotracer should be able to find it. Now we had to broaden the concept even more. Look at the many different compounds and see if one from the Optotracer family could be the molecule that can help us out. So we tried our prototypes, we tried what is sold under the name of Carbotrace 540, and we tried a few other development molecules. And from all these results, and comparing them to free xyloglucans, to reference celluloses, to our different treatments of the sample, we could exclusively show that we actually have cellulose. So what we can find in the end is that, yes, we do have cellulose in these Ulva algae. And the cellulose nanofibrils that we did isolate from Ulva is indeed high purity alpha cellulose. And the cellulose comes from a competing polysaccharide that is a copolymer of cellulose and glucose. And this is really a nice example that I would like to share with you on how Optotracer can help us to sort of solve tricky questions when working with unknown and quite complex matrices containing carbohydrates. So this is where we are now, taking this concept even further, digging deeper down into the structure of this algae and many others, and actually arriving to the dream that I showed you early on. This is what we want to be able to do, and this is what we actually can do now, thanks to the Optotracer technology. So we continue our work with algae. On the side and in parallel, we also continue our work with terrestrial plants, of course. There are so many byproducts from agriculture that stand out as feasible and sustainable alternatives to fossil oil for making materials. So we are working with different stools, byproducts from agriculture, and combining our conventional techniques that gives us a lot of information on morphology with the Optotracer technology to help us understand what they consist of, where and what molecules I can find in my biomass. for the most successful fractionation and recovery of material precursors. And with that said, I thank you so much for listening in on my story today. Thank you, and a heartfelt thank you, of course, to all collaborators and to AIMS. It's really such a joy to be part of this transdisciplinary research consortium, open-minded, creative, and fantastic people. Thank you all for listening today, and back to you, Ben.